Okay, so today I hope that we can get through all of DNA replication and start with DNA repair. So I just wanted to kind of summarize some of the concepts that we discussed so far in regards to DNA replication, right? Some of the questions that we wanted to address or at least be aware of as we go through the lectures was whether or not during DNA replication, the DNA strands pair with either the new or the old DNA, right? And if you guys recall, there was a really nice set of experiments done to determine whether or not the DNA is either pairing with the old strand or pairing with the new strands or together, right? So the conservative, semi-conservative, or dispersive type of DNA replication. So being familiar with those experiments that determine, that DNA was actually pairing with one of the old strands and the new strand was an old and a new strand is going to be important. Also, what proteins and other factors are required. Some of them we briefly touched on. We're going to get into more detail about the additional proteins in the lecture today. And we also talked about how and where does DNA replication begin, right? So the origin of replication, there's a single origin of replication, and whether or not replication go in one or both directions, right? So you have in bacteria, the circular plasmid. You also saw that the origin of replication opens and you have two replication forks. The DNA is being replicated bidirectionally. And what ensures that replication is precisely accurate, right? So we got into, I think we briefly touched on this, right? That the very last slide where we talked about the polymerases and how they have the ability to proofread to make sure that the correct base is being added. And we'll talk more today about how does DNA replication deal with supercoiling and tangling, right? As you're separating the strands of DNA. So just as a recap, we talked about the fact that in bacteria, there's 3 DNA polymerases. And each of these DNA polymerase has a different activity and a different role in DNA synthesis. So there's DNA polymerase 1, 2, and 3. All 3 of these polymerases have 5' to 3' polymerase activity. And all 3 of these polymerases also are able to have a proofreading ability, okay? So they can repair DNA in the 3' to 5' direction, right, for all of them. DNA polymerase 3 is the main polymerase that's involved in DNA replication. Polymerase 1 and 2 is involved in DNA repair. 2 is a little bit less known about what the original function is. So polymerase 1 is the one that's most often used and studied looking at DNA repair. And what's different also about these 3 polymerases is that polymerase 1 can also proofread or repair DNA in the 5' to 3' direction, okay? So as I said, all of these make DNA only in the 5' to 3' direction. They all require DNTPs. If you're going to do a reaction, so you need to have the nucleotides, the template, the primer, and a free 3' OH group to add those nucleotides to. The polymerases in eukaryotes is a little bit more complicated, right? There's much less known about the proteins that's involved in DNA replication. We know that there are multiple DNA polymerases that are involved, and we have some idea about the role of some of the polymerases in eukaryotes, okay? So they have different, depending on the type of polymerase it is, you have different roles throughout the DNA replication process. So some of them are involved in elongation of the lagging strand or the leading strand, DNA repair. Eukaryotes have mitochondrial DNA, right? So some are involved in replicating mitochondrial DNA. And also you can have some polymerases that might be involved in priming of replication of both strands. Okay. So very little is known about it. So we don't know as much about the eukaryotic DNA polymerases as we know about the ones in bacteria. So let's talk about a little bit more of the detailed mechanisms of what actually happens during DNA replication in bacteria. So at the start of DNA replication, what's important is having that DNA open to allow for DNA replication to occur, okay? And that initiation happens at the replication origin or the origin of replication. We know that that region is usually about 245 base pairs in length, but within that 245 base pair long origin of replication, there's important factors that are important for this initiation of replication to occur, okay? And that, that region includes 4 9-mers, right? So each of these blue boxes are 9 base pair in length. And then you have these 3 13-mers. So each of these red boxes are 13 base pairs in length, okay? So these are important features for that origin of replication. And at that origin of replication, the first process that starts is where you have a protein called DNA-A. This protein, with the help of ATPase and also another protein called HU protein, helps to bind to this 9-mer region, okay? So it recognizes these sequences, binds to these sequences, and when it binds to these sequences, it kind of changes the conformation of the DNA. So it bends the DNA, right? And you can imagine as you're bending the DNA, you can be forcing the DNA to become destabilized, right? So you might initiate breaking of bonds, etc. So binding of these protein to the DNA, these 9-mer region, bends the DNA. When this happens, you have an additional protein that comes in called DNA-B with the help of DNA-C, right? So these two protein complexes interact, and they're able to bind and recognize that 13-mer region that you see here, okay? It can then bind. It helps to actually open up this double-stranded DNA, right? So as you bend the DNA, bonds are broken, DNA gets separated into this little bubble, right? And we call this an open complex. When the DNA-B and DNA-C bind, DNA-B have helicase activity, so it will start helping to unwind the DNA as well, okay? So you see it's attached to DNA. And this unwinding then triggers this open complex, helps to recruit another protein called primase. And then primase can then bind and make those primers that you need to initiate DNA replication, right? Those little RNA primers. Okay, so it's kind of a stepwise process that this actually happens. And at this particular region of the origin of replication, having that open complex is absolutely required for DNA replication to occur. If there's no open complex, there's just no DNA replication that's going to happen. And this really requires that this DNA, with the help of ATP and also the HU protein, bind to this region. What's special about this region is that you do have this 13-mer sequence after the DNA-A binds, changes the conformation. This region, this open complex, is very easily able to separate the strands of DNA because it's highly AT rich, okay? So it has 81% AT. So AT, it has 2 double bonds, so it's much easier for you to break and open up those bonds, right? So it's not only that it has these 9-mers and 13-mers, you know, the composition of the nucleotides are also important to help to create this open complex of DNA. These two proteins, DNA-B and DNA-C, they together interact with each other. DNA-C helps to activate DNA-B, right, bringing it to bind to that 13-mer sequence. Because it has helicase activity, it will attach to both strands and literally pull them apart and help to unwind them further. And when this happens, you have another protein called single-stranded DNA binding protein, or SSB, which binds or coats that separated DNA strand to make sure that they don't come back together. Okay, otherwise they will close and the polymerase is not going to be able to access it. Primase is not going to be able to access it to synthesize those primers. Okay, so DNAB pulls it apart. These single-stranded binding proteins, as the name refers to, will bind to each of the single strands and prevent them from coming back together. Okay, as the DNAB continues to open the strands of DNA, it will continue to coat, and you can continue you can then create that open complex for DNA replication. What's also important is that this single-stranded binding protein has an affinity for DNA polymerase III. So in a way, it helps to recruit DNA polymerase III to that region as well. So once double helix is open, as I mentioned, right, you have your strand that are coated by the single-stranded binding protein. You need to make a primer to start DNA synthesis. And that primer is made by primase, right? These short RNA primer is made by primase. And then DNA polymerase can start extending into 3' in 5' to 3' orientation on both of these strands. These strands, which are then, the lagging strand here, will also have a primer made and can synthesize DNA in that lagging strand. What's interesting is that as you're unwinding the DNA, right, so you're pulling these two strands apart, the DNA is twisting. And you could imagine this again, if you have a string that's, interlocked with each other, and you try to separate those two string components, you can see that it kind of starts winding and creating kind of a tension. And that's exactly what happens with DNA. As you're unwinding these two strands, there's tension that's being formed on the double strand that's before that, okay? And the way that you need to relieve this strain, otherwise you will have some kind of tangling happening. And the way that you relieve this is with the help of these two proteins or two enzymes called topoisomerases. They are also referred to as DNA gyrases. And they help to relieve the strain on this double-stranded DNA as it's being unwound. In general, in bacteria, replication goes about, how quickly would you think that replication occur? Like how many nucleotides can be added in a certain amount of time? Or how many, I will give you kind of a hint. How many nucleotides do you estimate can be added or synthesized within 1 second? Just a random guess. 100? Thousands? Yeah, so we're kind of in the ballpark, right? So anywhere between 500 to 750 or even 1,000, right? I mean, that's kind of average. 1,000 nucleotides can be added per second, right? So it means that it's a very fast process, right? So you're really unwinding this DNA very rapidly. The two enzymes that helps to relieve that tension is topoisomerase 1 and topoisomerase 2. Topoisomerase 1 relaxes those supercoil by nicking and resealing one strand of the duplex DNA. Okay, topoisomerase 2 on the other hand relaxes supercoils by nicking and resealing both strands of the duplex DNA. So what that means, for example, if you have, if we're looking at topoisomerase 2, you have a strand, right? This is double strand kind of tension that's formed. It will cut this top strand, yeah, and pass it and I guess, relocate it. So it's cutting it, and then it's relocating it to the back. So now this strand is to the back of this twisted DNA. And by just resealing, cutting and resealing it relieves the tension, right? So you can see that this position is a little bit different, and it relieves the tension that way. And we can look at this more even further, right? If you are unwinding the DNA, replication machinery, as you're unwinding, it's twisting, it's forming these supercoils, as we refer to them. Topoisomerase 2 comes in, cleaves this double-stranded, both strands of the DNA, and replaces it, right? So it's going from this strand being behind, right, to it now being above, right? And that helps to quickly relieve whatever tension is there in the double-stranded DNA. So that's kind of the sole purpose of these two topoisomerases, is really to help to relieve the tension so that DNA replication the machinery doesn't stall, okay? So to put everything together, right, you have lots of proteins, and as I mentioned, what are the proteins involved? DNA polymerase 3, super important, right? That's the polymerase that's involved in DNA replication and synthesis. We also talked about DNA B, which is kind of the helicase. It helps to unwind the DNA. DNA G, or primase, either one of those names, helps to make that RNA primer that helps the polymerase to start synthesizing the new strand of DNA. And then you have these single-stranded binding proteins that really helps to keep the strands separated between each other, okay? And then you have your Ogasaki fragments. So you can imagine in an exam, you could be asked, what are these proteins? You could be asked a graphic to identify what are these proteins doing. So this slide really kind of summarizes important players that you should be aware of, okay? What's missing from this slide is topoisomerase, which will probably which would be upstream if it was bound to this DNA. It would be upstream, okay? So you want to think that topoisomerase is somewhere here. So how does DNA replication occur with these two different strands? We talked about that you have to have the lagging strand synthesized by forming these little primers that kind of jump ahead, right, of the DNA that's being synthesized. Leading strand has no problem, right? You can go 5' to 3', 5' to 3', and as the replication forks are being opened, you know, you have your leading strand going. These lagging strand need to be synthesized. So this is the 5' to 3' primer. You start synthesizing here. It will stop, right? It's going to stall here because you have this other piece of DNA that was synthesized. So the next, as this opens, you will have another piece, right, of primer here. It will get synthesized until it reaches this end. So that's why it's semi-discontinuous, right? Because this is continuous, this is just pieces being made. These pieces need to be fused together, right, to be able to fully replicate the full strand of the DNA for both of those strands. So these gaps need to be filled in. The enzyme that helps to fill in these gaps is really DNA polymerase 1 and DNA ligase, okay? So you guys recall that ligase is important for ligating pieces of DNA, and DNA polymerase is the one that actually helps to fill in these gaps, to synthesize these missing gaps that's there. And the way that these two proteins are able to do that is, you can imagine you have your double-stranded DNA, right? You have 5' to 3', you have your primer, the red part, this is the primer, right? That's there. You have this gap, right? Because this strand synthesizes and stalls right here. So you have a gap that's remaining. So what happens is that DNA polymerase can bind to this gap, because DNA polymerase, why is DNA polymerase 1 able to do this and not the other DNA polymerases?
Because it's able to work in both 5' to 3' and 3' to 5'.
Exactly, right? So it needs to be able to work at this 5' region of this primer. So it binds to that primer. It's before it's able to seal that gap. This DNA polymerase needs to remove this primer. So it will just start removing the primer, but also synthesizing new strands of DNA. So you can see that this is being extended along the way, right? So it's removing the primer and then continuing the synthesis as well, right? So in a way, it's doing both of repairing and also synthesizing those gaps. Once it gets to the end where it is, you have this nick, then DNA ligase comes in and seals this nick in this double-stranded DNA. Okay, so they work together to fill that in. The automated DNA polymerase 1 helps to, in this repair mechanism, or in the more of a proofreading capacity, is looking at bases that are incorrectly added, right? So if you have an incorrect base added, so here you should have had a C instead of an adenine is added. DNA polymerase recognizes that this adenine is there. This is the 3' direction. It uses the 3' to 5' exonuclease activity to remove this nucleotide so that the replication machinery can continue and it's no longer stalled. Okay, so it'll remove it and the DNA polymerase is able to continue the extension. So it's working to repair the gaps in that lagging strand that's left by the ogocyclic fragments and also that's left by the primers that are there when they're removed. But it also helps to proofread DNA that's being synthesized and if there's an incorrectly placed nucleotide as well. So, errors happen, right? I mean, this is just by random chance. So you can have random mutations either because maybe the wrong base is added and the polymerase is not able to repair it, so it gets incorporated, right? And that creates a mutation. It's a change in a base pair that wasn't there before. And that can create a problem for the DNA, okay? So again, we have a lot of cell division that's occurring in the normal human body in the course of a typical lifetime, right? Spontaneous mutations can happen depending on the DNA replication and also the repair mechanism not able to repair it. So you can have an estimated rate of 10 to the minus 6 mutations per gene per cell division, right? And if you were to translate that into every single gene over the course of a lifetime, it's quite possible that one of your genes in your genome would have undergone a mutation about 10 to the 10 separate occasions, right, throughout your life cycle. So mutations happen and they randomly occur. Is it possible for a human genome to go through? That's not possible. I mean, you'll see as we talk about DNA damage, we're exposed to so many possible things or environmental factors that can cause mutations. And also just the DNA replication machinery, polymerase, and the repair mechanism, they're just not able to do that precisely. So there's always going to be mutation just by random chance. With so many mutations, why are humans still living so strong? So a lot of mutations that happen don't necessarily happen in genes or important regions. A lot of mutations happen in regions that are not important in the human genome, right? You guys recall we have 3 million base pairs, 3,000 million base pairs within our genome, right? And only in that huge proportion of that genome, only about 30,000 genes. So there's a lot of regions in our genome that doesn't code for a gene or an important gene, right? So it's non-coding DNA. So a lot of mutations can happen there and they might not be detrimental, right? So we can live with it or we get fixed or anything, so we don't have to worry about it. So it's just the nature of how mutations happen. So this DNA polymerase do fix mutations anywhere in the strand wherever there's a mismatch. but its job is also to help to repair, to remove those primers from those Okazaki fragments and fill in that gap. Yeah, but it also does other, removing other mutations as well. So yeah, so we get back to this summary slide, right? So now you should be able to answer all of these questions, right? That we presented before based on all of the information that we just discussed. So as we're talking about mutations, right? You know, how do, you know, we have to go through DNA replication. How do we deal with the different types of mutations that, or change, or DNA damage, I must say, that happens within the cell? So when we think of DNA damage, we're thinking more in terms of a modification that's caused by a chemical alteration. This is different from if you're thinking about a mutation. So a mutation in general is a change in a basic, in a nucleotide that wasn't there, right? So you're changing the base pairing of DNA, right? And that's what creates a mutation. And because that change in the base pair could cause a change in the codons of the amino acid, right? And then your protein is not functioning properly. You could imagine that if you have certain modifications to a base within the genome, but it doesn't change necessarily the base pairing with another, with the adjacent or the complementary nucleotide, then it's just kind of a damage within the DNA, and that damage will get repaired, okay? If within the cell in general, right, you have like three kind of really common types of DNA damage that we often refer to. So you can have a base modification where the base gets modified by adding a particular agent or an alkylating agent, and we'll talk about what that is in a minute. You can also have pyrimidine dimers that's caused by radiation, and particularly UV radiation. And you can also have random double-stranded breaks, right? So these random double-stranded breaks that just happens, right, within the genome because of exposure to a chemical or something else within the genome needs to also be repaired, okay? So I think I've updated these slides, so we'll skip over this. I don't think those are the slides we posted. I was doing another presentation, so let's skip over this slide. And I'll update that in the lecture. So DNA damage that's caused by alkylation of the base is really because of the composition of DNA, okay? So there's some substances that's present within our environment that are referred to as electrophiles, right? Or they're electrophilic, meaning that they love negatively charged electrons, right? So when they encounter these negatively charged electrons, they will attack them, okay? Because of the composition of DNA, you have this phosphate that's fusing these two bases together, right? This phosphate, sugar phosphate backbone that have this phosphodiester bond. And there's a negatively charged molecule here, electron here. So certain chemicals in the environment recognize this negatively charged group and attacks it, okay? And it will modify the base in this way. When it attacks this base, it will add a carbon-containing group, which is an alkyl group, and that's why it's referred to as alkylation, right? Because it's adding this alkyl group, so it's referred to as alkylation. In addition to this oxygen that's present on this phosphorus that's negatively charged, you can also have other regions that can be attacked as well on the actual bases of the DNA, right? So N3 and N7 are very common regions that get attacked by these electrophiles. But there's also all these other nitrogen that's shown here and oxygen that's shown here in blue can also be attacked, but not as much as you would find the oxygen here and also the nitrogen 3 and nitrogen 7 that's being attacked. what happens is that when you have these alkylation of these particular bases, depending on where it's occurring, it can be either harmful or harmless, okay? So one of the common type of alkylation that can happen, so for instance, we're looking at guanine here, right? So if you were to have this N7 alkylation of this guanine, it doesn't do anything, right? It can still base pair with this cytosine, right? So it's modified, there's an alkylation, the DNA is considered to be damaged, but it doesn't do anything to the base pairing with the cytosine, okay? However, alkylation of N3 is a little bit more serious, because what it does is that when this gets alkylated by adding one of the alkyl groups to it, it cannot base pair properly, right? Here with this thymine, and it also cannot base pair with any other base that's going to be coming in to be recognized here. So when that happens, it's considered to be a non-coding base, because it doesn't, the DNA doesn't know, the polymerase does not know which base to add to it, okay? So could be considered a problem, and that needs to be fixed, okay, or repaired in some way. In terms of the O6, which is kind of the one that's the most obvious target for these electrophiles, and that's the one that leads to the most mutation, is this O6 modification. Even though it's not commonly attacked, it's very rarely would you find that this O6 is actually attacked, but it's the one that causes the most problems, I should say, okay? And the reason is because it actually changes the base pairing during the next round of DNA replication. And what I mean by that is that you have a guanine and the cytosine. What happens, a alkylating agent such as EMS, right, or ethyl methane sulfonate, comes in, adds this metal group to this O6. When it adds this metal group to this O6, this pairing that you would normally have, this third bond that's normally hydrogen bond that's formed between guanine and cytosine, cannot happen anymore, right? So this is really a problem, because then the polymerase doesn't know that this was originally a guanine, right? So what it recognizes that you have these two base pairs to pair to. So instead, a thymine is added, okay? Because this thymine gets added during the next round of replication, this thymine will pair with an adenine, right? So you're going from a G to C original base pairing, and that gets switched quickly to an A to a T base pairing. So you're changing the base pair. This is a problem, of course, because depending on if it's in a coding sequence, you're changing the codon composition. The protein is no longer, it's going to probably have some kind of mutation, and a different protein is going to be made, or a truncated protein, or some modifications in a protein is going to be made. So EMS mutagenesis quite often causes very problematic mutations within the cell, okay? There's many other types of environmental carcinogens, such as similar to EMS, that's present within the environment, and they can also cause mutations, right? This is just an example of a few of those mutations that often occur, okay? And one such as well environmental factor is UV radiation, right? You know that UV radiation, it has a biological significance to all of us, right? Because it's present in sunlight. So most pretty much all organisms that's exposed to sunlight to some extent is exposed to UV radiation. And UV radiation can cause a mutation, and that's why for some people, excessive exposure to sunlight can cause skin cancer, okay? Because you're really damaging the cells and the division of those cells, okay? And UV, the components within sunlight or UV radiation, it just damages the DNA in the cells, and that leads to a mutation, okay? And then you have an uncontrollable amount of cell division that can then lead to cancer, okay? So UV radiation is, again, it's low energy, so it's not as high as gamma radiation, but it's low energy. What it does is that it results in pyrimidine dimers, right? So you have your double-stranded DNA exposed to UV radiation. You have these two thymines that form, these two pyrimidines that form these dimers, okay? So then these bonds are broken here, and the bases are not able to pair with these two pyrimidines anymore. And they're often called CPDs, or cyclobutane pyrimidine dimers, because it forms these four carbon ring here, okay, to form that dimer. And of course, it will block DNA replication because it's non-informative. The DNA polymerase doesn't recognize a pyrimidine dimer, so it doesn't know what to do with it. Okay, so it will definitely block DNA replication. It also distorts, right, the structure of the DNA duplex, right? If you guys recall, in the last lecture, I mentioned that the spacing between the bases are kind of important for DNA polymerase as well, right? So it really distorts the DNA in addition to being non-informative to the DNA polymerase for replication. The other type of radiation that can damage the DNA is a more energetic form, which is gamma and X-rays, right? They can interact directly with the DNA molecule. They can ionize the water molecules that's around the DNA, right? So if there's water molecules that's present around the DNA, they can ionize them. And they form these highly reactive free radicals that attacks the DNA, okay? It attacks the DNA in different parts of the DNA, alters the bases. It can also just break the double-stranded DNA. And the damage to the double-stranded DNA, the damage to the DNA can be either just by breaking just a single strand or a double strand, okay? So these are also highly, as you can imagine, highly problematic within the cell that can lead to cell death as well if they're not repaired properly, okay? And a lot of these mutations, again, they're removed before DNA replication. That way, when the new DNA gets replicated, it's already repaired, right? But some of them are not. So the way that you fix or repair DNA damage, it depends on the type of DNA damage that we're dealing with, right? So one way to fix it is to repair it or restore it to the original state, okay? And the two ways that this is usually done is by undoing the damage or removing the section of the DNA that was damaged and then filling it in with new undamaged DNA. Okay, so one example of undoing the damage is usually involve a light reaction or light repair. And this is it's called photoreaction because it requires light, okay? And if you have, as remember, UV radiation causes these pyrimidine dimers. There's an enzyme called DNA photolyase that recognizes these pyrimidine dimers. This enzyme is able to recognize these pyrimidine dimers, bind to these pyrimidine dimers. Because this enzyme absorbs light, and that's where the light repair comes from or is referred to, it absorbs light and gets activated. The activation of this enzyme, then breaks these pyrimidine dimers, and these dimers are then able to base pair with the original nucleotides in the double strand. And then this enzyme gets released, recirculates in the genome or in the cell, and wait for the next wait to encounter the next pyrimidine dimer to repair it. The other type of mechanism to now repair a more high energy DNA damage, right, is using a enzyme that's referred to as a suicide enzyme. Why it's referred to as a suicide enzyme is because after it does its job, it kills itself. So it can't be used again, right? So, I mean, so it's kind of like, it's costly, right? It's costly for that enzyme. In a way, it's just there to really help to repair damaged DNA, and that's really its sole purpose, right? It cannot be used for anything else after. When a lot of enzymes have multiple functions, this one just really have one particular function, and is to repair the damaged DNA caused by high energy irradiation or exposure, and repair that base. So O6 alkylation, right, on the guanine, and that's the one which actually converts the DNA into from, you know, either a GC to an AT. These type of modifications or damage is recognized by this enzyme which is referred to as O6 methylguanine methyltransferase. Okay, and if you just refer to this, you know, in exams or whatever as the suicide enzyme, that's perfectly okay, because it's a very long name. But what this enzyme does is that it removes this methyl group by adding the methyl group to the sulfur within that enzyme, this sulfur atom, and removes it from that location. When it adds, when this methyl group gets added to this sulfur atom in this enzyme, then that enzyme gets destroyed, and it's no longer able to be functional, so it's inactivated. And this is really just a mechanism to repairing these high energy types of DNA damage. And so you're repairing it, right? So you repair it either by a light reaction, right, using the photolyase, where you break the pyrimidine dimer. You remove it by using the suicide enzyme to remove that methyl group from that modified guanine. But you can also repair the DNA damage by removing completely the damaged DNA and not necessarily the component that was there. And there's two ways to repair DNA damage in this way. One is by doing what we refer to as excision repair, right? It's the process where you remove most of the damaged nucleotides and replace it with fresh DNA, okay? And there's two types of that depending on how extensive that damage might be. So you can either have a base or a nucleotide excision repair mechanism. And they're often referred to as BER or NER for nucleotide excision repair or base excision repair mechanism. Base excision repair is more prevalent and usually works on, you know, very common or small or subtle changes to the DNA base, right? So it can even help with some of the chemical modifications. Nuclear excision repair is definitely more costly, right? It requires more energy, and they deal with much more drastic changes in DNA. So DNA that might, so changes to the DNA bases that might distort the DNA and become a problem within the cell. It can help to repair as well damage such as pyrimidine dimers, right? It can also function to repairing pyrimidine dimers as well. And the way that this works for the base excision repair, again, many different, for each of these particular mechanisms, you have a particular enzyme that's, you know, very specific to that particular mechanism, right? So that's the job of these enzymes. And the enzyme that's important for base excision repair is DNA glycosylase. And this DNA glycosylase recognizes this mismatch, right? This damaged DNA, right? So you have the incorrect base, right? Or damaged base that's shown in red here or in pink. And this DNA glycosylase will recognize it, invert it or switch it from the inner region of the strand of DNA to the outer region so it can be removed, right? So then it cleaves it and removes it, okay? Because you're removing the base, what you're left with is just that sugar. So you're removing that base, and this remaining part that's present within the double-stranded DNA is usually referred to as either apurinic or apyrimidinic, okay, right? Because either you're removing the base, so it's no longer have the purine or the pyrimidine, depending on what it is, okay? So this region is then recognized by particular endonucleases, right? So the endonuclease will come in. It will then cut this DNA at the 5' end of the site. So DNA is always written 5' to 3'. So it will cleave at the 5' end, right? So the site is here, so it will cleave at that 5' end of that site. And that cleaver, this cleavage is then further removed by another enzyme called DNA phosphodiesterase, which comes in and removes that sugar that's remaining here, okay? Because the nucleotide is gone, this is the sugar backbone, so that gets removed, and that's what you have here. And then DNA polymerase 1 comes in, like it does with all repair, and fills in that gap and extend along the DNA, and DNA ligase will seal whatever nick is remaining behind, okay? So it's a combination of finding the base, getting rid of it, cleaving the region around it, remove it, synthesizing a new strand with new undamaged DNA, and then sealing that with DNA ligase. It's just kind of a stepwise process. In a more extensive DNA damage that's more bulky, right? So such as, for example, I said pyrimidine dimers, right? Which you cannot just easily remove by using one of the other types of mechanism to remove it. You will do the cell will then use nucleotide excision repair mechanism to remove this damage. This involves XI nuclease, which actually comes in and cuts 12 to 13 nucleotides, right, on either side of the damaged DNA, right? So you're removing a huge chunk of this DNA, and it removes it. DNA polymerase 1 and ligase, right, will come in and synthesize the new strand of DNA, right? So this gets removed, the new strand of DNA. So this helps to repair the DNA that way. So depending on the nature of the DNA damage and or the base change that's present within the DNA damage, and how extensive it is, right? You will have either using base excision repair or nucleotide excision repair. On the other hand, you might have a double-stranded break, right? And double-stranded breaks are incredibly problematic, right? Because they're the most dangerous form of DNA damage. These are usually they're broken chromosomes, right? Because they're broken chromosomes within the cell, if they're not repaired, of course, it can lead to cell death. And they can also lead to cancer in mammals, right? So in vertebrates. If for some reason you're not able to fix the these double-stranded break, the cell will quickly go into apoptosis, which is cell death, right? So fixing it is important. So you have mechanisms, more than one mechanism to deal with those type of double-stranded break. And these mechanisms will also function depending on where in the cell cycle these events might be happening, okay?
Professor?
Yes.
You say that in the vertebrates it can lead to cancer. Is that after it's attempted these ways to fix it, or is this before or during?
So it's depending on where in the cell cycle this damage is occurring. So for example, if you have your DNA that's damaged, you have double-stranded DNA, let's say it's not fixed, the replication machinery stalls, right? So because the replication machinery stalls and that cell might continue to divide uncontrollably, making more and more of these broken chromosomes, and then that eventually leads to cell death or to cancer cells, right? So it really depends on where, particularly in the cell cycle, that this is happening, right? Many times, if these damages occur before the DNA has replicated, then they will get fixed prior to DNA replication. If it's happening after the DNA gets replicated or during that time, they're really more of a problem then, because that's when it kind of stalls everything and create a problem.
Okay, thank you.
And in the way that the cell deal with these double-stranded break, right, is either to repair it using what we refer to as homologous recombination, right, instead of one of the many different mechanisms, because it's a double-stranded break. It's not a base pair change, right? So base pair change, you can use either the nuclear, the NER or the BER, right? Or you can also, depending if it's a DNA damage, you can use either the light reaction in that case to fix this, right? But it's a double-stranded break. How do you deal with that? Homologous recombination or non-homologous end joining. And both of those mechanisms will help to repair that broken double-stranded DNA. So if we were to look at non-homologous end joining, for example, right? This particular method actually alters the original DNA sequence when you're repairing this broken double-stranded DNA. The reason being is that when this double-stranded DNA gets broken, the repair process requires that you process some of the bases adjacent to that double-stranded break. So by processing it before you start repairing it, you can lose some of the bases that are present, okay? So it's not a perfect mechanism for repairing double-stranded break because you really lose some of your DNA sequence using this repair mechanism, okay? So this type of repair mechanism is usually used when the duplicated DNA have not, or when the DNA have not yet duplicated their DNA, okay? So this happens mostly in somatic cells, for example, right? Another problem with this mechanism is that the cell does not have a way to prevent this double-stranded break and this piece of the double-stranded DNA from fusing to another piece of DNA that's on another chromosome, okay? So because of this, right, because it's just putting two pieces together, because of this, you can have a lot of rearrangements, right? Where one chromosome that's broken can be attached to another chromosome. And this actually can happen, and it happens, right? So our genome have, a lot of times you will find it in our genome, chromosomal rearrangements, and that's because of these double-stranded break, and they're just coming back together randomly, okay? The other mechanism to repair double-stranded break is homologous recombination, right? Where the broken sister chromatid gets repaired by using the information from the sister chromatid, okay? So even though you're processing the ends, right, because you need to process the ends to seal, it can fill in these gaps using the sister chromatid. So the sequences get preserved. So you, in this way, you don't really lose any of the DNA sequence as you would do here, right? So this repair mechanism is definitely more accurate in that sense. This mechanism is very similar to recombination in bacteria, right? So recombination repair in bacteria. This type of mechanism as well takes place after DNA has been duplicated, okay? So this one is before the DNA has been duplicated, right? Mostly in somatic cells. This one occurs after the DNA has been duplicated because you need to be able to have this duplicated template DNA before the next cell has divided, right? So if you guys remember the cell cycle, you duplicate your DNA, then you go through the cell division part, right, during mitosis or meiosis. So this happens at a very particular time, or it has to happen at a very particular time in order for you to be the cell to undergo homologous recombination. Otherwise, that's a problem that doesn't get repaired. What would lead to double-stranded breaks? Again, just double-stranded breaks can also occur from just random environment chemicals in the environment or carcinogens in the environment. You can have random double-strand break. Or you can have, perhaps there's some nick within the DNA that occurs within the cell randomly, and somehow it doesn't get repaired. And that leads to breakage of the other strand of DNA, but most times it's really just double-stranded breaks that's formed from environmental factors, especially strong high energy environmental factors such as gamma radiation, etc. So you can have these double-stranded breaks that way. So for non-homologous end joining, right, again, this is a stepwise process. Don't get bogged down too much by the name of the proteins. The idea being that if you have non-homologous end joining, the ends are broken. You have these proteins which are referred to as Ku protein, which is a DNA-dependent ATPase. So it has, it can phosphorylate, it functions as phosphorylation enzyme, right? So these proteins bind to the broken DNA to protect it from actually being destroyed further or being cleaved further or destroyed by nucleases, because we always have nucleases around in our genome, right? So it kind of protects it from being further damaged. When it binds to the ends of these DNA, together with this DNA protein kinase, it helps to bring these two strands or pull these two strands together. The pulling of these two strands together also, with the help of the helicase activity, so Ku also have helicase activity, so it unwinds just a little bit of this double strand that you're going to be pulling together, and it needs to unwind it because you need to be able to base pair them together to fuse them together. Okay, so it's pulling them together, unwinding it a little bit so that you can fuse them together. When you fuse the two bases together, or whatever matching bases you can have that will be fused together, whatever is remaining gets removed, gets cleaved from these two strands that's pulled together. And gets filled in by DNA polymerase and ligase to seal that broken DNA, okay? So this is kind of a very basic model for non-homologous end joining. It's a little bit more complicated, but this is more of a simplistic view, right? It's really pulling the two strands together to bring them, to be able to seal them and put them back together. The other, for homologous recombination, that's also a very complicated process. So what I want you guys to focus on is the fact that you're using the sister chromatin to be able to do this, right? It is the preferred method for repairing double-stranded breaks because you're not losing any DNA by doing that. You're also avoiding chromosomal rearrangements, right? So in this process, this happens shortly after the DNA has been replicated so that you have this, these two sister chromatids still within, within the vicinity of each other, okay? Double-strand break, you have nuclease that helps to digest the ends, right? And create these overhangs. This broken strand, the 3' end of this broken strand, invades the sister chromatin, yeah, and uses that sister chromatin as the template to synthesize this gap. Okay, so this all is green is being synthesized here. When this happens, after this happens, this gets filled in, and then the top strand will fill in using this as a template. Okay, so it's kind of a strand invasion to synthesize the double-stranded break, the strand of double-stranded break, and then it gets sealed in by ligase. Okay, it's accurately repaired. Both of these are very they're homologous, they're sister chromatids, so they're identical. So it gets repaired. You don't lose any DNA. Okay,
Professor.
Yes.
Why is it called nuclease, not endonuclease?
So yeah, it's probably it's more than likely an endonuclease as well. Yeah, but it just uses nuclease. Yeah, it's probably an endo. It's more than it has to be an endonuclease. Yeah. But nuclease is kind of a general term that they refer to as for enzymes that cleaves either the end or outside or inside. It's just a general term, and then you can specify it by endo or exonuclease, but it will probably be an endonuclease here. So naturally, homologous recombination happens, right? Within our cells, right? Because we have, if you guys remember from genetics, we often have novel combinations of our genes, yeah? And this is really a deliberate action within the cell, right? Is to have this homologous recombination occurring during meiosis so we can exchange the material between different chromosomes, okay? And this is really just an example. Do not get bogged down by all of these protein names, right? Knowing that this happens to generate chromosomal crossover so that we can recombine and have different sets of combinations within our chromosomes. So the process is very similar to what I mentioned earlier, right, for homologous recombination. So, these are the proteins, right? These nucleus is complex, right? These two different proteins that comes processes the ends of this DNA, this broken DNA, similar to what I just showed you. So you have this strand invasion that's happening, right? Similar like how you would repair a double-stranded break. The strand invades, and once this strand invades and you start synthesizing this new DNA, you can have one of two things happening, okay? You can have what I just told you, where you have the chromosome, where you have the DNA being repaired and repaired perfectly, right? So it's repaired, there's no crossover. So this is very analogous to this process, right? However, you can have an alternative mechanism that's happening, right? Where you have crossover, and this is where the recombination comes in, and this is where the shuffling comes in as well, right? So very similarly, it goes through this step, synthesizes the new DNA that's being made, and actually it doesn't let go of the strand that it's used, the strand that it's invaded. Okay, so the strand is made, and instead of these two being released, you have what we call a recombination event happening, right? So this and this region gets recombined. Okay, so you're going from your top strand, which is normally, but then you also have this red strand that's here taking up this part, right, of this DNA. Okay, so this type of recombination forcing these crossover events happen, and they get, depending on how they get resolved, right, because you imagine that you have this twist in the DNA, they need to be resolved, right, and depending on how they're resolved will determine the type of recombination and rearrangements that you, or the type of recombination events that you have. Okay, this kind of crossover event is referred to as a Holliday junction, right? And as I mentioned, depending on how this gets resolved, will give you two different combinations. This is a very straightforward, right? So you have that crossover event, and I'm showing you a single crossover event here, right? Not a double one. And if it's cleaved right here, you have your top strand, right? Red, blue, and then you have red. So the ends are preserved with the original chromosome, right? So heteroduplex or patch recombinants, okay? So the ends are really preserved, so you're just taking a piece of it. Here, which is more what we refer to as splice recombinants or crossover recombinants, is where the ends are no longer preserved. So one end is the original red or blue, and the other end is the other one, coming from the other chromosome. And that's if you're cleaving vertically here, right, at 3 and 4, position 3 and 4. Okay, so you'll be cleaving, you're maintaining your top strand, but then you're using the piece that's here, okay, for this blue strand. So depending on which one it is, that's really what you need to remember, depending on how you cut this. And this is resolving these different damaged DNA or broken DNA within the cell. This is great. This happens, of course, in eukaryotes. DNA replication in bacteria, very straightforward. The problem that we have with eukaryotic chromosomes in terms of DNA replication is that they're not circular, right? We have linear chromosomes. And because we have linear chromosomes, the ends of those chromosomes are very susceptible to being destroyed or shortened or attacked, okay? So they need to be protected, okay? So because it's linear, you need to be able to distinguish normal chromosome double-stranded breaks from the ends of chromosomes, right? So as I mentioned, if you have a double-stranded break, one of those break might pair with another chromosome because, again, there's two ends together, right? The system doesn't recognize that they're two different things, right? So sometimes these chromosome breaks can result in end-to-end fusions of different chromosomes. And this one, the end, will lead to chromosome breakage and cell death, right? Because, again, you're breaking off the chromosomes into pieces and fusing it with something else. The way the DNA replication work, right, with our leading and lagging strand, you have these RNA primers that needs to be removed, okay, at the ends, at one or two ends of the machinery or of the DNA that's being replicated, okay? Because you have to get rid of the RNA primer, you can imagine that this can lead to shortening of the ends of the DNA. The ends of the DNA, you guys all know, is referred to as telomeres. So you can really shorten the telomeres on a particular chromosome every time you go through DNA replication, right? Because every time you go through DNA replication, you're having this 5' primer added, and that 5' primer needs to be removed, okay? And this is really to explain what I just said, right? You have this linear chromosome. At some point, you're gonna have these two strands separated, right? They're linear, right? Your leading strand is fine, your lagging strand, you have these primers that are present. These primers gets removed, and usually the primer gets filled in because DNA polymerase will come in, right? DNA polymerase 1, remove this primer and continue synthesizing along here. The problem here is that DNA polymerase can come in, remove this primer, this RNA primer, but it doesn't have a 3' OH to continue extending, right? So you're left with this shortened piece of DNA here, okay? It doesn't get repaired. During the next round of DNA replication, right, these two will get separated. You have a new strand, it's shorter, right? One is shorter. So you can imagine that you keep shortening and shortening your DNA if there isn't a way to fix this. Luckily there is, right? So work by Elizabeth Blackburn, who's at UC Berkeley, in the 1980s showed that the ends of our chromosomes, which are telomeres, are protected by the addition of additional sequences using an enzyme called telomerase. And because of her discovery of how these chromosomes are protected by telomeres and telomerase, she won the Nobel Prize for this in 2009. Now we know, again, a lot more about how this mechanism works in many other organisms. And what it is at the end of our chromosomes, what gets added are these repetitive sequences, which are highly GC rich or G rich, right, in the strands. And it's added to this 3' end at the end of every chromosome, okay? The telomeric sequence in humans versus other vertebrates is usually a known sequence or common sequence or consensus sequence, you refer to them, right? So TTA, GGG. And this can be repeated for a few thousand base pairs. It all depends on the type of cell, and we'll talk about that in a minute, okay? So the 3' end of these G-rich strand can extend 12 to 16 nucleotides beyond the 5' end of the complementary C strand, right? And that way it helps to preserve that end. That's where the primer gets removed. This region is bound by specific proteins that protects the end of the linear chromosomes from attack by exon nucleases, right? So these are nucleases that are floating around in our cell that is looking to chew up any type of broken DNA or ends of DNA for that matter. So telomerase is the protein that's involved for to add these telomeres or these sequences to the end of our chromosomes. And it adds these sequences because within the telomerase, the enzymatic activity of this telomerase is very similar to reverse transcriptase. Okay, and you guys remember you saw reverse transcriptase when we were talking about QRT-PCR, right? Reverse transcriptase PCR. And you guys remember what we would use reverse transcriptase PCR for?
To convert RNA to DNA.
Exactly, right? So you're converting your RNA to complementary DNA or cDNA. And this telomerase have reverse transcriptase activity, and it carries its own internal RNA template to direct this DNA synthesis, right? Because you remember with DNA replication, you make an RNA primer, right? So primase comes in, makes this RNA primer, you can use that to continue the synthesis of the DNA. Here, this telomerase actually carries its own RNA primer that can then recognize that DNA sequence or just start synthesizing DNA, this G-rich DNA at the ends of the chromosome, okay? So if I ask you, like, what is the composition of telomerase, the idea is that you'll tell me that the composition of telomerase is this reverse transcriptase and this RNA, okay, that's present. The process for adding these telomerase or these telomeres to the ends of chromosome is kind of, it's a stepwise process, right? And I'm not going to go through all of the details here, but I'll focus more on the figure that we have over here, right? So you can imagine here, this is the end of your chromosome, the 5', and this is the 3' end, right? That you will be adding these GC-rich regions to. And this telomerase enzyme, the pink one, this is the RNA. You can tell it's RNA because it has U, right, instead of T. And it comes in base pairs with just 3 bases, right? So TTG, it can just base pair with just 3 bases. The rest of it gets annealed, right, to this region. And it just needs to find this region to start. Once it recognizes this region, it slides along the DNA, shifts the position, keeping these 3 bases connected, right, to keep it attached to the DNA. And then these strands get synthesized, okay? 5' to 3' get synthesized here, okay? And it can keep sliding along, finding this, so this next TTG, it binds here, synthesize, the next TTG, it binds there, synthesize, right? And it keeps doing this, you know, depending for how long. There's no real limit to this, right? So we'll keep extending the ends of these chromosomes. Of course, you'll see it too much is a problem, right? And once it gets to a certain distance, right? Here is just showing you for demonstration purposes, it's here. It leaves the strand of the DNA. Primase comes in, right? So the primase will now be able to add a primer here, okay? So once it's sufficiently long, primer comes in, makes an RNA primer to DNA, right? Because it has something to attach to now. And it will synthesize the rest of, DNA polymerase will come in and synthesize the rest of this strand of the DNA, right? And fills it in. So that way you have these ends that get extended to the end of your chromosomes, right? And the top strand also is going to be using, is going to be synthesized as well, okay? So you protect the ends of the DNA this way, really preventing it from being shortened after each round of DNA replication. Does it mean that this is foolproof? You guys know from maybe from at some point in one of your classes, in one of your science classes, as we age, right, our telomeres shorten as well, right? So, you know, over and over time, right, your telomeres can shorten. You can also have your telomeres being shortened under certain types of stress conditions or certain types of diseases. So quite often we like to make fun and say, you know, if we're really stressed out about something, our telomeres are getting shorter and shorter, right? And this naturally happens. Whether or not this is due to the telomerase becoming less efficient or less active, it's not really clear, but it can happen, okay? But in general, telomeres are there to maintain the ends of our chromosome and prevents it from being shortened and fused to something else, okay? So, and depending on which cell we're looking at, right, you will have different length of telomerase being added, or telomeres being added, or the different lengths of telomeres, or those repeated regions at the end of your chromosomes. So embryonic cells, germline cells, and stem cells, right, they produce telomerase. That's where you get it from. And only, in somatic cells, you only have a very low number of telomerase. And this is usually as they enter the S phase of the cell division cycle. And because of that, they have a low telomerase. Their telomeres shorten with each cell division, right? It's pretty low, yeah. Sometimes you can have a complete loss of telomeres. And if you have the complete loss, we end up with this situation we talked about double-stranded breaks, right? You can have end-to-end chromosome fusion because there's nothing at the ends to protect this from happening, okay? And extensive shortening of telomeres can be recognized in the cell as a kind of DNA damage. And this will lead to p53 triggered apoptosis, yeah? And you have cell death. Okay, and I'll stop on this slide, but I just wanted to say I was going to get through this particular slide and summarize. So just bear with me for 1 or 2 minutes if you don't mind. So here again, you're having chromosome, the end of the chromosome. Depending on the cell that we're looking at, right, you can have different combinations of the telomeric length. So embryonic cells or stem cells, you can have 3 to 20 kb, and this is 20,000 base pairs of this repeated sequence, right? Indefinite replication, cells are long. A reduced number of these repeat at the end, right? You have low telomerase, you don't have as many, limited replication in the somatic cells. You have, you can have shorter chromosomes this way, or shorter chromosomes this way. If you have ends with no telomeres, right? Again, there's senescence, and senescence just means dead or old or aging. And this can be a problem for the cell. You have fusion chromosomes being fused together, leading to cell death, right? It's highly unstable. And in cancer cells where you have 55,000 base pairs of this repeated sequence, again, because cancer cells really continue to divide a lot, right? So it's uncontrollable growth and cell division. You can also lead to chromosome instability, breakage, and deletion because it's really out of control in terms of the number of telomeres you have to the end of these chromosomes. So just to briefly summarize, and then we'll continue in the next class with the rest of the slides. DNA can be damaged and then repaired, and can even be mutated beyond repair, right? You can have recombination, homologous recombination, if you're repairing a double-strand break, but you also have forced homologous recombination during meiosis. And eukaryotic chromosome, because they're linear, the ends need to be protected, and they're protected by addition of these repeated sequence towards the end by the enzyme telomerase. So this is just to summarize before we continue on transposons and the next lecture. So that's where I will stop today. And I'll stick around for you guys if you guys have any questions.
Thank you, Professor.
Thank you. Yes.
Can you go back like 3 slides? Yeah, here. So when you look at the strand at the bottom, the top one is still shorter like by 3 nucleotides.
This
Or
one?
3, basically. The very bottom one.
This one?
You go all the way down, yes, yeah. The top strand is still shorter like by 3 nucleotides.
Yeah, it's still shorter here, but this is the end of the chromosome. So all of this is additional sequence, right? So this doesn't really matter because you already have all of this already protecting the end here of this chromosome.
Right, but I mean the whole purpose of this process is to keep the telomeres, right? But the telomeres are now shorter, still shorter.
The purpose here is to protect the ends of the DNA by adding telomere sequences. And the length of these telomere sequences will vary depending on what it is. This schematic here is really just showing you an example. Usually it's much longer than that, right? As I mentioned. So this is just to demonstrate how telomerase actually work. But this is not like a real life scenario where you have this situation, because most cells you have, 3,000 to 20,000 base pair repeated sequence. So this is just really just demonstrating how this actually.
Yeah, that makes sense.
So this is the
Yeah,
this will be long, like way beyond this end of this chromosome, right? And it just
Mhm, mhm. Oh, thank you.
Mhm.